Thank you. Thank you very much. I want to thank very especially Dr. De Robertis and Monsignor Sanchez Orondo for this kind invitation. And I'm also very grateful because in this day and a half, I have learned a lifetime of science. So let me give you an idea of the magnitude of cardiovascular disease, especially stroke. And here we have work that was done in this academy a few years ago that we entitled Atherosclerosis, the 21st Century Epidemic. And you can see in this graphic very easily that you have all the causes of death in the world, infectious disease here, accidents, cancer, but the main cause easily seen on the graphic is vascular disease with stroke, a myocardial infarction, diabetes, and infarction. And indeed, if you have a rank of deaths in the world, myocardial infarction is #1 and has been #1 since 2005, but has increased 50% in these years until now. And stroke, sadly, is increasing to the 4th position, increasing 20%, whereas infectious diseases like diarrhea have decreased. So is it a priority? Well, yes, of 160,000 deaths that occur in the world for every reason every day, a third are caused from vascular disease. And stroke ranks 1st, 2nd, and 3rd cause of death in the world. In the United States, it used to be 4th cause, and now it's 3rd cause again. It's in Argentina, Brazil is 1st or 2nd cause of death. There are 15 million people affected by strokes in the world per year, and of those, 6 million die, and 5 million have sequela, making stroke the #1 cause of disability worldwide. Risk factors, epidemiologically speaking, of course, somebody who is overweight, has a father who died young, has diabetes and smokes 3 packs per day, has a very high risk of stroke or cardiovascular disease. But epidemiologically speaking, most of vascular events, 80%, occur in people with few risk factors that are not expecting to have a stroke. All risk factors for stroke affect all race and ethnicities similarly, so there are no differences. And strokes and MIs are due to the same risk factors in all the world. A large study in almost 50 nations showed that risk factors for cardiovascular disease are always undertreated, and Feigen mentioned that there is moderate geographic variation, so we should think that it should be easy to fight cardiovascular disease. Well, that hasn't been the case. The developing world is even worse because of all the strokes, 87% occur in our regions. That is like 13 million strokes. Are we prepared to deal with this? No. And the prevalence of stroke in our regions is lower, but that's not good news because it's lower because we have a higher case fatality rate. That means people who have a stroke die due to poor health services. In developing regions, strokes and cardiovascular disease occur 1 to 2 decades earlier compared to developed countries. That is, 50% of people affected are younger than 70 years of age, compared to 20% in developed regions. And of course, the most vulnerable are the ones that know the least. In our stroke unit, there is no patient or family that knows or can name a symptom of stroke. But we are missing an opportunity because every reference I can show you, and there is one by WHO there, shows that just by doing what we know, diet, exercise, no smoking, using the drugs that we have available, we could decrease vascular disease by 80%, and it's not happening. So let's look at this data from the Danish. They have electronic records for the entire 5 million population from the Queen down, and they did a meta-analysis comparing 90,000 people who had vascular checkups like the ones you had to prevent cancer and vascular disease, and 90,000 people that did not have health checkups. And what was the difference between the two groups? None. There was no decrease in death in people that had checkups compared to those that did not have them. So why is that? Because of this. Here are 2 real cases of young men, 48, 47, that if you measure their risk based on population-based scores, like the Framingham score, here you have one that smoked, both have bad cholesterol elevated, one had hypertension, the other one diabetes, the other one excess weight, and according to the population-based risk score, they both have low vascular risk, but we applied an individualized approach in which we measured arterial age or accelerated arterial aging by measuring vascular rigidity and plaque area. Pulse wave velocity reflects rigidity, which is independent of plaque load, and it was elevated on this patient, and he had a lot of plaque, 60 square millimeters. The baseline for that age should be 10 to 15 millimeters. So we reclassify these patients to a very high stroke and a myocardial infarction risk on this one, and a normal risk for the second one when we use this individualized approach. And here we have then the population-based medicine, which is good for governments, in which you can use a polypill with 5 different drugs, but you are going to over-treat some people, and you are not going to treat enough some others at very high risk with this approach. We have used the personalized approach that I just mentioned using and measuring arterial age, and we published last year a study on 600 people measuring plaque load, in which in 80% we changed statin medication based on the plaque they had in their arteries. But the next step is precision medicine. Can we move forward using precision medicine and its derivatives? Well, in stroke we have a problem. The problem was well put by Kaplan in an editorial in the 80s when he said, what is wrong with Mr. Jones? Because people with stroke come with a hemiplegia or blind or not speaking, but the underlying etiology is very different. Stroke is a very heterogeneous disease. Look at this graphic. You have 2 ends of a spectrum. Hemorrhagic, 15%, where a blood vessel ruptures, and 85% is occlusion. Large vessel disease, like the carotid, where you can have a partial occlusion throwing emboli to the brain, or you can have a total occlusion producing a hemodynamic deficit. And this large vessel in the neck is different from large vessel inside the brain, in the middle cerebral artery. Then you have small vessel disease, affecting vessels that are less than 1 millimeter in diameter, causing the so-called lacunar infarcts. The heart can be a cause of stroke with atrial fibrillation, which is highly embologenic and not always easy to find, but treatable with anticoagulation. And then there are rare causes like dissection, prothrombotic states, migraine, and others. And then we have a large group, almost 20%, 25% of strokes are cryptogenic or ESOS, embolic stroke of unknown origin, where we can't find the cause that left that patient hemiplegic. So these could be the answers. So genomic medicine. Now we have computers that can analyze these 3 billion pair of bases and find if we have the clue to understand prediction and treatment of cerebrovascular disease. Obama in 2015 proposed that genomics and molecular biology should be used in daily medical practice. And the NIH went forward with this, starting an initiative with a cohort program, just to study better the genotypes of people with correlation with the diseases. But the problem is that stroke is not a monogenic stroke. Less than 7% of strokes are monogenic. And one example is CADASIL, the dominant form, and CARASIL, recessive, which is in notch 3 of chromosome 19, and occurs in people with migraine with aura who have strokes at age 50 that lead to dementia. There is no treatment for this disease, but genetic counseling could be useful, and there you have the image of the white matter severe disease in people with CADASIL. Another example is Fabry disease, a lysosomal storage disorder that has a deficiency of alpha-galactosidase due to a mutation in the GLA gene, and there are more than 170 mutations described, and it does have a treatment, a new treatment, which is intravenous galactidase A and B, which treats the skin manifestations, the kidney insufficiency, and the very painful polyneuropathy associated with Fabry, but not the strokes associated with Fabry, unfortunately. A very example of monogenic diseases are cardiovascular ones, where they have a single locus and a gene with a major effect, and they cause syncope, or very commonly they manifest with sudden death in young people. Long QT syndrome, short QT syndrome, Brugada, all EKG alterations with multiple mutations that show during sports, especially swimming, more in boys. It used to be thought that it could occur only in athletes, but then it was found that it could occur during sleep with emotional or auditory stimuli. So it's not just telling somebody don't do sports to avoid death from this. And also hypertrophic cardiomyopathy that you cannot find at autopsy, so you need molecular biology to diagnose all these common causes of sudden death. This is the most common sudden death cause in young athletes, 12 genes, almost 1,000 mutations. And a very important paper by Van Driest, which showed that in 2,000 people, he found mutations for these disorders in 200, but there were no manifestations, no EKG changes, no symptoms. So he warned about giving diagnosis of potentially lethal diseases to people that overestimating gene penetrance that will not manifest in a phenotype. So let's go from genotype to phenotype instead of the other way around. And the idea that payers will cover this genetic testing, they avoid doing it. And the reason is that these affect a very small percentage of the population. Sudden death is an excellent example. In the general population, very few people have a sudden death. If you go to somebody who had a myocardial infarction with a stent, with arrhythmia, of course it's a greater percentage. But when you go to absolute numbers, of course, this small percentage in the general population is a huge number. So payment and coverage for these gene tests is definitely justified. And familial hypercholesterolemia is one great example. This is the receptor for cholesterol. This is the LDL, bad cholesterol molecule, which is bound to apolipoprotein, forming lipoprotein A, and it binds to this receptor that takes the LDL particle into the tissue, taking it out from the blood. But there is this enzyme here, PCSK9, that destroys the receptor and increases LDL in blood, and this is Brown-Gall's disease, more common in people of Jewish ancestry, that results in severely increased LDL levels of 400. There are multiple gene mutations. The receptor is the most common. There are recessive ones, and it increases 100% the risk of early death in the second decade of life. So here we see a few trials of statins that, as a side, they were studied genetically. There were genetic studies done during these trials, not as the main endpoint. But a genetic risk score was done with 57 DNA variants, and this polygenic risk score was applied, and it was interesting that the people in the top quintile of the risk score had an associated 10% for each standard deviation increase in the risk score. There was a 10% increase in plaque burden. And if you take only the WASCOP study, for the same LDL, the relative risk reduction of vascular events was 44% for the people with the genetic risk score versus 24% of those that did not have the genetic risk score. And for all the studies combined, the absolute risk reduction in vascular endpoints was 3.6% using a genetic risk code versus 1.3% if you did not have the risk score. Let's see the impact of familial hypercholesterolemia. Here you have a homozygous person. This is the LDL level in the green line that leads to vascular events. When you are homozygous, you can start having vascular events at age 15. If you don't have familial hypercholesterolemia, you may have starting events at age 50. And when you are heterozygous, when you start having events will depend on whether you are treated soon enough or not. Statins are drugs that made a major difference in the last 30 years in survival, probably for humans. And the LPA gene predicts this much better than measuring lipoprotein A in the blood, because the number of polymorphisms of the Kringle4 type 2 in the gene defines the size of the LDL particle. And we know now that small LDL particles are much more pathogenic than larger ones. But that's not measured commonly in laboratories, and it should be done. ApoE4, known for Alzheimer's, decreases survival in myocardial infarction, but that is reversed by statin treatment independent of lipid levels. Coenzyme Q10. Many patients are afraid of taking statins, and many physicians are against statins because of muscular side effects. And that's a serious, very serious mistake. Less than 5% of the population could have some side effects, and this is because the statins work on the mevalonate pathway, decreasing isoprenoids, among them ubiquinone, which is coenzyme Q10, and we have improved some discomfort in muscle by giving supplements of coenzyme Q10 in patients. It's the cytochrome P450 in the liver that defines 10 to 50% of the variation in response to statins, although there are some mutations, like in this gene for simvastatin, but hydrophilic statins like pravastatin and rosuvastatin are the best tolerated and the most widely used. PCKS9, I mentioned before, the increases plasma LDL by destroying the receptor, but of course there is a loss of function mutation which reduces cardiovascular mortality, and drugs have been used, evolucumab, just published in the New England Journal, showing that it decreased the vascular endpoints very significantly, reaching LDL levels of 30, which was never thought it was possible before. So stroke is a polygenic disease, very heterogeneous. You are not going to find one DNA mutation explaining stroke, because we showed before genome-wide association studies are likely to show risk scores to find causes for or association, increasing risk for stroke. Thousands have been published so far, and there are some genes that have been identified in this meta-stroke analysis collaboration with 60,000 controls and 12,000 patients that define cardioembolic stroke, probably because of an increase in the risk of atrial fibrillation. There is also another gene, HDA-C9, which has an inhibitor that could treat large vessel disease, that in a combination of 40 genes, you could make a difference between cardioembolic and large vessel disease stroke, which is very important at the emergency room, because you have a very narrow therapeutic window to treat stroke, about 4 hours, so you need to know the mechanism to do the best treatment possible. The GRECOS project is a project to make a model for prediction of recurrency of stroke. The highest risk for a stroke is to have a stroke. So these candidate genes were studied, 1,500 patients that had an average of 6% recurrence of stroke in 1 year, but the MGP gene related to artery calcification increased the hazard ratio, and patients were reclassified from a low risk of recurrence at 2% to a very high, almost 13% risk of recurrence if this gene is identified. Pharmacogenomics, clopidogrel has been shown, 25% of people are resistant to clopidogrel, which is a drug used when a stent is placed. The problem is that if you're resistant, you have a 3 times increased chance of stent occlusion. So the mutation should be studied in people at risk, because stent occlusion is associated with a 50% mortality rate. We wrote to the president, actually, Macri, whose father had open heart surgery at age 53. Macri is 58 now, and he should have that mutation done, of course, and most people should have it done in case they need to receive clopidogrel. There is also aspirin resistant, and the SNPs and genes associated with aspirin resistance, which cannot be measured in vitro, have been also identified. And of course, anticoagulation. Everybody knows that having somebody on a therapeutic range with warfarin is very difficult. It was attributed to eating vegetables high in vitamin K, spinach, broccoli, cauliflower, etc. But now we know that there are actually mutations that explain the resistance. And here, there is loss of function in clopidogrel, but there is also gain of function. If you have 2 alleles of gain of function for clopidogrel, you go to the other extreme. You can have a high rate, double rate of hemorrhage by using clopidogrel. Vulnerable plaque. In 1995, a paper was published discussing the vulnerable plaque, and this is a very important concept, not widely known, in which an artery in a segment which is not stenotic, actually it has plaque in the wall, but this is the most likely to cause an infarction, because that little plaque that does not occlude the artery can rupture, that's the vulnerability, and cause the myocardial infarction. 86% of myocardial infarctions and strokes occur in arteries that are not narrowed and cannot be detected with a stress test, ergometry, etc. The infarcts associated with arteries that are almost occluded is just 14%. So we are doing now studies, genomic studies in endarterectomy specimens to compare arteries that are stable. Unfortunately, most people that have a carotid endarterectomy, they have not had symptoms. They have asymptomatic stenosis, which should not be operated, but 90% are operated. So we have specimens from people that have stable plaques, and we are comparing them to the people that have surgery in unstable plaques who had symptoms to try to find what defines an unstable one plaque from the other, considering that most humans from age 30 on have plaques in their arteries. So what defines a vulnerable plaque? There are many things found with GWAS, 2 sites that increase intima media thickness, variants in these 2 genes that show greater plaque load and instability of those plaques, which is reversed by omega-3 fatty acids in fish, not in capsules. The OCT4 gene, very important because it decreases the necrotic core of a plaque, it decreases the hemorrhagic complications of a plaque, and it increases the thickness of the fibrous cap, making the plaque much more stable. And finally, RNA expression analysis that identifies plaques that are not stenotic but are vulnerable to occlusion. So we have this incredible amount of complex data that is generated through high throughput biotechnologies nowadays. So the question is, how are we going? This is the limiting factor here. How are they going to, the 5 million researchers in the world, how are they going to process this data to make new discoveries, drug discoveries, or treatments? There are 70,000 bioinformatics software tools now available in the web. There were 30 new ones per week in 2013, 200 now. So this is more exponential than Moore's law. How do we handle this? In conclusion, we have to identify the variables of unknown significance. As I said before, mutations without phenotypes, phenotypes without mutation, changing research from genotype to phenotypes and not the other way around. Risk stratification, genetic risk scores for a polygenic disease, a stroke, randomized clinical trials using these genetic risk scores, they should help not only to prevent sudden death, but also MI and stroke prevention in general. The vulnerable plaque, again, everybody has a plaque, and most plaques do not need to be stenotic to cause an infarction. So which are the ones that are more prone to cause those infarctions? Familial hypercholesterolemia in children, when do we test them? How do we treat them? Should we use the PCSK9 inhibitors now? Pharmacogenomics. All drugs, of course, the oncology field is much more advanced than any other field, but there are many drugs now that have these companion diagnostics to know about their effectiveness and their side effects. They should be used much more broadly. Epigenetics, I have not mentioned, but it's crucial. Nutrigenomics, folate, hypomethylation, that will be discussed later, I guess, and all the omics derived from genomics. This is James Gandolfini, a famous actor that received a prize in Sicily, and actually he died while receiving the prize at 51. His son found him in the bathroom. This happens every day in thousands of patients. This shouldn't be a surprise. The problem is that we need physicians' education to translate your research into clinical practice. To us, I feel like a second grader babbling these words that we are not used to use, and most of the terms are not understood by physicians in clinical practice. So there is a large work to be done for physician education. And, well, yeah, Churchill used to say this, health is a transient state which does not predict anything good. Well, we should define what health really means and be able to say who is healthy and who is at risk. Thank you.